All right, scientists, let's take a look at topic 8 of the IB Biology higher level syllabus. We're going to start with subtopic 8.1, metabolism. And it would be great if you had a solid knowledge of enzymes because this subtopic relies heavily on your knowledge of enzymes. So let's get started. The metabolic pathways consist of chains and cycles of enzyme-catalyzed reactions. So as you know, enzymes are tiny machines. They are proteins most of the time that catalyze reactions. They speed up chemical reactions in your body. So your metabolism, the building and destruction of molecules, also known as anabolism and catabolism, all these reactions are what make you alive. Life is a result of all these zillions of chemical reactions that happen all the time, and they need to be fast. Those reactions need to happen fast because if a certain product is required for, I don't know, respiration, for example, and you don't have that product readily available, then respiration will not be possible. And then you don't have the energy to stay alive. To make sure that those reactions are fast and you always have what you need, the enzymes will catalyze chemical reactions. They will speed up chemical reactions. So this metabolic pathway could be a linear pathway, could be a chain of events. Just like a factory, you have an assembly line, you have the raw product, and then after several steps, you end up with your finished product. Here in this linear metabolic pathway, you start with a substrate, and after a series of reactions mediated by different enzymes, you end up with your product. Now, it's important to remember that each enzyme, each reaction is catalyzed by a different enzyme because enzymes are very specific. Enzymes are specific for substrate. They are specific for each reaction. So here we have 3 enzymes. We start with this substrate, and then the product of the first reaction is the substrate for the next one, right? So this is what makes it linear. So start with a start with a substrate, and then after a series of reactions, you end up with this finished product here, and it goes on to be used by the cell in some other process, right? So it's a straight line. Now, a cyclic pathway, you also have different enzymes doing different reactions, but in a cyclic pathway, the end product here is the input again for the first reaction. Now, it might be the case that reaction from A to B also releases carbon dioxide, and reaction D to A requires the input of this other molecule, E, for example, that is combined with D to form A. Not many pathways are cyclical, but 2 of them are very important, the cycle of Krebs and the cycle of Calvin. They are part of respiration, of aerobic respiration and photosynthesis, respectively. And we are going to visit them in subtopics 8.2 and 8.3. Now, enzymes lower the activation energy of chemical reactions that they catalyze. What does that mean? Every chemical reaction requires an activation energy. Now, think of, for example, the combustion of paper. You have paper as a fuel plus oxygen, and that will give you carbon dioxide and water, right? This is the combustion of paper. You have cellulose on paper, which is a polysaccharide. Combining that with oxygen will give you carbon dioxide and water, and heat energy will be released. So when energy is released, we call it an exothermic reaction. But why isn't, I don't know, toilet paper in your bathroom right now? You have paper, you have oxygen in the atmosphere. Why isn't the reaction happening? Why isn't your toilet paper on fire right now? Well, hopefully your toilet paper is not on fire, because to start the reaction, you need to add some energy. So here we have on this side here, an example of exothermic reaction just like this one, right? So the reactants have higher energy than the products. So the reactants have higher energy than the products, and this difference is the energy being released. For example, in the combustion, you feel the heat, you see the light. All this is the energy, the difference between the reactants and the products. But to go from reactants to products, you gotta go through this hump here. You gotta go uphill first, and then you can release all this energy. And the energy required to go over this hill is called activation energy. In the case of paper, you might need to light a match first to give this first energy to start the reaction. When you light a match, you are rubbing the matchstick on the box, and this rubbing is adding energy, right? This friction will add energy to the matchstick and will promote, this reaction here. In your body, all sorts of exothermic reaction will happen. For example, glycolysis is not one single reaction, but it's an exothermic process, will release energy, right? You will end up with ATP. You will end up with NADH. Where did this energy come from? It comes from consuming glucose. But you also need to overcome this hill, this energetic hill. Think of an actual hill. To make a ball, for example, go from this point to this point here, you have a higher height to a lower height. So this point has more potential energy, and this point has lower potential energy. It should be a smooth ride, but to go over the hill, you have to add some energy, and then all this energy is released. Same thing here, you have more potential energy, chemical potential energy. And here you have less chemical energy. So you have to add some energy here to go over this hill and then down. For endothermic reactions that also happen in your body, same thing is true, except the reactants here have less energy than the products. So endo means towards the inside. So endothermic means the energy is going inside the molecules. The energy is going into the chemical bonds to make the products. So anabolic reactions, for example, they are endothermic because you're going to take many monomers, for example, monomers of amino acids. And when you make a protein, you have to consume energy. You're going to consume ATP to just synthesize your protein. Why do you need energy for that? Not just to overcome the activation energy, but also because the products have higher energy than the reactants. So you need this energy in order to complete this endothermic reaction. What do enzymes have to do with that? Well, the enzymes will lower the activation energy. So let's draw step by step this graph here. Let's say I have my axis. So I have my energy here and the reaction progress, so the time here. In both situations, I will start, let's do here an exothermic. This one is endothermic because it's absorbing energy. Let's show an example of an exothermic. The products are here with a higher energy and they require this activation energy. Sorry, these are the reactants. The reactants start with higher energy. They require an activation energy, and then you have the products. So all this is the activation energy. What enzymes do is they start from the same reactants and they go to the same products, but they take a shortcut. They take a shortcut. So the activation energy for an enzyme catalyzed reaction is much lower. And since you don't need as much energy, reactions can happen much faster, right? How do enzymes do that? They can stabilize the molecules. They can stabilize the intermediate step in the reaction. They can orient the molecules in the best position. They can put everything in place so that reaction can happen faster. So by improving orientation and stability of the molecules, they can lower the activation energy and make the reactions faster. Now, there are some molecules called enzyme inhibitors that will, exactly as the name suggests, inhibit the enzyme activity. So the enzymatic activity, the rate of reaction will be lower in the presence of an enzyme inhibitor. There are two types of inhibitor. One of them is competitive inhibitor, the competitive inhibition. As I am sure you remember, the enzymes have this place here, this part of the molecule called active site. And the active site is where magic happens. The active site is the place where the substrate can enter, can connect to the enzyme in the active site, and that is where the reaction happens. But a competitive inhibitor is a molecule that may look like a substrate. They might have a similar shape, they might have a similar size, and they also have an affinity for the enzyme. They also like the active site of the enzyme. So the inhibitor will plug in the active site and block the active site. And then the substrate cannot attach to the enzyme. They cannot form a complex. And because of that, it's as if you had fewer enzymes. The inhibitor will compete with the substrate for the active site. Some inhibitors are covalently bond to the enzyme, so they are permanent inhibitors. We call that irreversible inhibition. But some inhibitors are not covalently bond. They just take the place and then they leave and they compete all the time with the substrate to see who takes the active site the longest time, who stays in the active site the longest. And when you plot a graph of the rates of reaction, this is what you get. Now let's dissect this graph. Let's understand what's going on here. Here we have V, and V is the rate of reaction. Think of V for velocity, if you will. So the rate of reaction, how fast the reaction is happening. And at some point, you will have the V max, the maximum rate of reaction. On the x-axis, we have concentration of substrate. Concentration of substrate. So as the concentration of substrate increases, the rate of reaction also increases up to a certain point where it plateaus. You have this plateau here. That means the reaction will not get any faster no matter how much more substrate you get. This is because all the enzymes you have in solution, all the enzymes you have in your assay are working at their full speed. And if you just pile up more substrate, that will not make them any faster. The thing is, if you add the inhibitor, you can see that the rate of reaction, the increasing rate of reaction is not as steep. It will eventually reach Vmax. It will eventually be as fast as the uninhibited molecule, as the uninhibited enzyme. And why is that? Because you have so much substrate that your inhibitor will be outnumbered by a lot, no matter how much affinity they have. If you have enough substrate crowded with substrate, then the probability of the active site to be occupied by one of those substrate molecules is very high. That means the rate of reaction, even with the inhibitor, will be as high as an uninhibited molecule, as an uninhibited enzyme. Now, how much substrate do you need to reach this level will depend on your inhibitor. So what is this Km here? This is a constant of affinity. This measures the affinity of the substrate for the enzyme. And it is measured by how much substrate you need to reach half of Vmax. So because of the inhibition, you need more substrate to reach Vmax, to reach half of the Vmax. So your Km here, the affinity is different. For your competitive inhibition, Vmax is the same. Vmax, even of the inhibited here, the Vmax is still the same, but Km is not. The affinity is not. One example here is captopril on the angiotensin converting enzyme. It's an enzyme that converts angiotensin 1 into, it is related to blood pressure, and captopril is a drug that is used in hypertension. So when the patient has a high blood pressure, high arterial pressure, you can use captopril to inhibit this enzyme that produces angiotensin 2 that promotes a higher blood pressure. Now here we have the ACE here in orange and green, and captopril is this purple molecule here in the middle. The helices, the spiral thingies, are alpha helices. If you remember, enzymes are proteins, and the tertiary structure of the protein is this 3D thing we see. The secondary structure are the alpha helices and the pleated sheet. So here we see that this enzyme is made of helices, and the green parts here are not helices. They're just like chain of amino acids between the helices. Now, besides competitive inhibition, you have non-competitive inhibition. You might also see the name allosteric inhibition. So this happens when the inhibitor connects to an allosteric site, a site that is not the active site, right? It's going to bind to this allosteric site and change the shape of the active site. Now, if you remember the lock and key model of enzyme activity, enzyme specificity, each key requires a different lock. So each substrate requires a different enzyme because they fit. They are right for each other. Even though we know because of the induced fit model, the enzyme changes in the presence of substrate, still the active site is pretty much specific for the substrate. And if you change the active site, then the substrate doesn't fit anymore. And then you block the reaction. You will inhibit the reaction because the enzyme, the shape of the enzyme is not favorable anymore. And the non-competitive inhibitor will do that not because it blocks the active site, but because it binds to the allosteric site. This will be the allosteric site. The inhibitor binds to an allosteric site, changes the active site, and then the rates of reaction heavily decrease. So look how different this graph is from the previous one we saw. Here, the Vmax of the uninhibited enzyme is not the same as the Vmax of the inhibited enzyme. It doesn't matter how much more substrate you add, the reaction will never be as fast because it's not a problem of the active site being occupied by an inhibitor. The problem is the active site is modified. The active site is not as good anymore. The substrate cannot bind to the active site anymore. So the rate of reaction will be lower. The maximum rate of reaction will be lower than the uninhibited situation. On the other hand, the affinity constant here is the same, both for the uninhibited and for the inhibited situations. So what is the difference when you look at the graph between the competitive and the non-competitive inhibition? competitive inhibition. Vmax is the same, but Km is different. Non-competitive inhibition, the Vmax is different, but the Km is the same. And an example we can give for non-competitive inhibition is alpha amanitin, which is found on death cap, the mushroom. It is a non-competitive allosteric inhibitor of RNA polymerase. So this here in cyan is RNA polymerase. In red, we have the alpha amanitin from the mushroom death cap. And here we see again alpha helices, even a beta sheet here. Look at that, a pleated sheet. So you have pleated sheets as well, and helices. Now, this is not the active site. This is not the active site. But when alpha amanitin binds to this site, it will mess up with some helices here, and that will impair the rate of reaction, the rate of synthesis of RNA, and that can have a bunch of consequences to the organism. Now, the metabolic pathways can be controlled by end product inhibition. It's a special case of allosteric inhibition. It's quite clever negative feedback control of production. Imagine you are making cupcakes. So you have a series of steps in your cupcake production. When you have enough cupcakes, you will stop making the cupcakes, right? You will stop not at the last step of the process, but you will stop making cupcakes from the beginning. There's no point in making the dough, etc., 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 and just stop at the very last part. So if you have enough cupcakes, you will stop the first step in the cupcake making process. So end product inhibition is when the last molecule, when the last product of your pathway is an allosteric inhibitor of the first enzyme. It's when the last product of your chain is a non-competitive inhibitor of your first enzyme, of the first step. And it's very clever because if you have enough of the end product, then you don't have to make more. On the other hand, if you don't have enough, then you keep making. One example that the IB will require you to know is the end product inhibition of the pathway that converts threonine to isoleucine. So isoleucine is an amino acid, so is threonine. Isoleucine is an essential amino acid. That means humans cannot make them. So humans cannot make isoleucine, but plants can. And the first enzyme, the first step is catalyzed by this enzyme, threonine dehydratase, because it removes here this. Your book might bring it as threonine deaminase because it will remove the amino group here, and that's the same, right? It's the same enzyme, two names here, threonine dehydratase, threonine deaminase, potato, same enzyme. And this enzyme is regulated, the activity of this enzyme is regulated by the last product of the whole pathway. So the end product, isoleucine, is an allosteric inhibitor of the threonine deaminase, the first enzyme of the pathway. Meaning if you have a lot of isoleucine, you don't have to make any more. So isoleucine will inhibit threonine deaminase, threonine dehydratase. But if you don't have enough isoleucine, then you do not have anyone inhibiting the enzyme and the pathway carries on. Skills you need to have to calculate and plot rates of reaction from raw experimental results. Now, this is a virtual experiment. The link for the website where you can download the software is in the comments. I picked one of the enzymes and I ran 4 trials. And then I took the average of those 4 trials and I plotted in a graph. Now my graph here has time in minutes, but usually we're going to have the time in seconds. So I converted here in this table. This is measuring the rate of production, the rate of formation of the product. This is an enzyme. This is just like this abstract enzyme. I think it's called enzyme 3 or something. They don't tell what product it is. They don't tell what enzyme it is. It's just for simulations. But you can measure the rate of reaction by measuring the formation of product or the consumption of substrate. You can measure, here is measured in micromoles, but you can measure, for example, the mass. It's just fine. You can measure the formation, the increase in product mass or decrease in substrate mass. If your reaction, if your product is a gas, you can count bubbles, for example. If the formation of bubbles, if your product, I don't know, changes color, you can use a spectrometer to measure the increase of the product using a spectrometer, using this device that will quantify the formation of this colored product or the destruction, the consumption of a colored substrate. So I plotted everything here. And by the end of 50 minutes on 4 trials, the average amount of product I had was 5.68 micromoles per 3,000 seconds. So the rate of reaction will be 5.68 micromoles per 3,000 seconds. So my rate of reaction will be 1.89 times 10 to the power -3 micromole per second. Per second. You can also express that as in times second to the -1, right? So this is the unit. Do not forget the unit. In this case is micromoles because this is what I'm measuring. If your reaction, I don't know, if you're measuring the mass of product or the mass of substrate, you're going to use the mass unit, but it's going to be per second, right? Per second. And the use of databases to identify potential new anti-malarial drugs. So here we have this image is from a blood slide. You see here the red blood cells. They have this disc shape. The center is lighter than the edge because the red blood cell is a disc with a depression in the center, right? So this depression makes the center here look more pale. And these circles with a dot, they are a parasite. They are a protozoan called Plasmodium. I'm going to call it Plasmodium sp because there are 4 species of Plasmodium that cause malaria. And the cycle of the disease is if a mosquito, which has the Plasmodium, bites you, the Plasmodium will enter your bloodstream. It will infect your red blood cells. They will multiply. They will reproduce asexually in your red blood cells up to the point where there are so many of them inside the red blood cell that the cell just pops. It bursts open like that movie Alien. It bursts open. Oh my God. And all these parasites come out to infect new cells. Usually, this will take 3 days to happen. There is one species of Plasmodium that takes 4 days. And then when this happens, because of all the intracellular contents are now spilled, you will have a fever. Your body will identify the damage and will develop a fever, to try and kill the pathogen. But the plasmodium will infect many other red blood cells. And 3 days later, boom, everyone will rupture again, and you will have another fever. You will have a fever every 3 days. You have a fever, 3 days later, another fever, and you're good 3 days later. And then you have another fever until the number of red blood cells is too low. And what do the red blood cells do? They carry oxygen. So if you do not have if you do not have enough red blood cells, then you do not have enough oxygen, right? And when animals do not have enough oxygen, they have this bad habit of dying, which is not good for your health. So we need to solve this problem. Luckily, there is a drug called chloroquine, and chloroquine is used to treat malaria successfully in many tropical countries. The problem is there are strains of Plasmodium that are resistant to chloroquine. That means we have to develop a new drug. But developing drugs is expensive. It requires a huge investment from the pharmaceutical companies. And then let's say you develop this new anti-malarial drug, and after a while, a new resistant strain comes. You have to develop yet another. That's a lot of work. It's a lot of money. It's a lot of problems. Luckily, there are databases. There are available databases with proteins, thousands of proteins that you find in the Plasmodium, for example. Or you have the genome of Plasmodium also available in databases. And you can use that, and you can use computation simulation to try different drugs. You can model different drugs. You can look for allosteric sites of inhibition. You can look for many things that are effective, and you're going to know if they are effective in the simulation even before you synthesize the molecule. So that saves a lot of time and a lot of money. That makes the development of drugs much easier. It's still a lot of process. It's still a very difficult task, but it's cheaper and faster than it used to be. So here we have, for example, from an actual science paper, you have this drug called mefloquine, a derivative of chloroquine, and you can see the simulations here in a ribosome. You can see the molecule interacting in the ribosome. You can see the active site and everything, the target site of the drug. And scientists, by sharing these databases, they can collaborate. You have a group of scientists that is responsible for, proteomics and developing these protein models, and they will help build several work groups will help to build these protein databases. So other scientists responsible for the medicinal chemistry, for example, will develop the drug models and see if they interact. And you have global cooperation of many scientists towards the development of new drugs.